Hello, class. So now that you're comfortable discussing the dual nature of our immune response, let's start talking about antigens. So antigens are antibody generators. They are things that generate your body, trigger your body to start making antibodies. So antigens are going to be found on pathogenic things. So these are substances that produce your body to make antibodies. Typically, antigens are going to be proteins or large polysaccharides. Typically, they need to be large. If they're smaller, they usually have to be grouped together in some form. And microbes, an antigen might include part of their capsule, their cell wall, their flagella, fimbriae, a toxin, virus coats, surfaces of any sort of pathogen. However, our immune system doesn't always do a perfect job, and sometimes we will start making antibodies in response to something that's not pathogenic or not microbial. And this will mean you'll end up developing an immune response. So some common antigens that are not pathogenic would include pollen, egg whites, blood cell surface molecules. So as a health provider, if you are giving someone a blood transfusion, you need to make sure that their blood types match. Because if they have a blood cell surface molecule, an A blood or a B type blood that doesn't match them, they'll develop an immune response and attack that blood. Serum proteins from other people or species and transplanted tissues and organs. So not only is blood labeled with different antigens, but tissues and organs also can have different proteins and polysaccharides on them that if you are doing a transplant, you'll want to match that transplant from the donor and the recipient as closely as possible to minimize the chance of their immune system rejecting that donated tissue. And often they'll also end up on and some sort of immune suppressant that will limit their ability to have their immune system attack their transplant. Antigens typically, so we said they're big, but it's typically not the whole big thing that your immune system is going to interact with. They'll just have little parts of them that are called an epitope. And that epitope is a small region of the antigen where the antibody is going to bind. So I feel like at this point it's fair game to say antigens, antibodies, words that sound very similar but are not the same. Antigens are the things on the pathogen that start your immune response, and antibodies are the things in your body that are the immune response. If there's something that is low molecular weight, it's called a hapten. And sometimes haptens are used in vaccines, but they have to be conjugated to a carrier molecule because these smaller molecules all by themselves are not going to be very antigenic. They're not going to trigger the body to start making antibodies. But by putting them on this carrier molecule, you have this conjugated molecule that then is antigenic. It becomes large enough for your body to start mounting an immune response to it. Antibodies are also called immunoglobulins. That globulin because they're globular, and immuno because it's part of the immune system. There are always proteins, and they're relatively soluble, which is important. That means it can flow through your blood. It can go through your body fluids. Antibodies are made in response to an antigen, and they are made to recognize and bind to the antigen. They have two identical sites that bind to the epitope. These are called the antigen binding sites. Valence refers to how many antigen binding sites are found on an antibody, and most human antibodies will be bivalent. They have a valence of 2. So if we go through, I would really like you to be able to draw an antibody from memory and label all these different parts of it. So there are 4 chains, 2 of these blue chains, 2 light chains, and 2 of the green chains, the heavy chains. So they're called heavy and light because the heavy ones are bigger, they're heavier, and the lighter ones they're smaller, they're lighter, so they're the light chains. And all of these chains are bound together. See these sulfurs here in the middle? Those are called disulfide bonds. So those disulfide bonds hold these 4 chains together. At the end of them is a variable region. This is also called the antigen binding site. So because they are variable, each one is different, and it's able to bind to different antigens. It's able to bind to different epitopes. So here you have a picture of, here's part of the antigen binding site, and here is the antigen. It's able to just bind to, because it just fits together. It's just a shape. The shapes fit. And then you have a stem, and the stem is a constant region. There are 5 different classes of antibodies, and each class has its own particular type of stem. Those 5 classes are IgG, IgM, IgA, IgD, and IgE. So if we go through these, there are some important things you need to be able to know about each type of antibody. IgG is about 80% of the antibodies you have in your body. So it is the big majority of your antibodies. And it is very important in phagocytosis. It's important in neutralizing toxins and viruses. And it protects the fetus and the newborn. In a lot of our immune response, IgG is the main one that's going to be acting in that immune response. IgM exists as a pentamer. So see how it has 5 of them stuck together? That means now it has a valence of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that's 10, a valence of 10. So it's really good at agglutinating antigens because it can grab 10 things and stick them together in one little ball all by itself. IgM is also one of the first antibodies produced in response to an infection. So typically the very first thing that we see is a rise of IgM when you're fighting an infection, and then it'll switch to IgG after that. IgA forms a dimer. You can see it's 2 pieces stuck together. This is found in a lot of body secretions. So just think of any types of body fluids. Your tears, your saliva, your mucus, your intestines, breast milk, blood, lymphatic fluid. And so because it's in these wet parts, it's important in protecting the wet parts of your body. It is going to give you a lot of protection on your mucosal surfaces. Because it's found in breast milk, it also can be transferred to a baby. And it is very important in helping prevent diarrhea in newborns. However, that baby is going to digest that like any other protein. So IgA won't make it to the baby's immune system. It won't flow through their own bloodstream. But it can protect against infections along the intestinal tract. IgD is a monomer that is found all over the surface of B cells. And so it helps in the functions of B cells. But there's not much is known about what it's doing in serum. And then IgE is important and found. IgE can bind to mast cells and basophils. And so, as you would imagine, it's involved in things that mast cells and basophils do. Allergic reactions and lysis of parasitic worms. So now you can tell me what part of an antibody reacts with the epitope of an antigen and which class of antibody is most likely to protect you from a common cold. All right, we're going to continue our talk on Chapter 17 in our next video. Let me know if you have any questions. You can email me or stop by office hours. And I'm happy to talk to you anytime you need. See you later. Bye.